Welcome students back to chapter 3. We're going to continue on with part 1. This will focus on section 3.3, which is about passive membrane transport. So our plasma membrane is our kind of selectively permeable barrier, right? So here is the plasma membrane. You see here's the kind of intracellular fluid, all the cytoplasm. And then out here is our external environment where we have extracellular fluid or interstitial fluid. So there are two ways in which stuff can move into and out of our cells across our plasma membrane. We have active transport and passive transport. So passive transport processes don't require energy, right? They're passive. In that instance, the substance that's moving will move from where it has a high concentration to where it has a low concentration, i.e. it moves down its concentration gradient. Now, active processes, right, they're active, they're energetic, so they will require energy, usually in the form of ATP, or at least indirectly always in the form of ATP. Active processes can move stuff against their concentration gradient, i.e. from an area where they have a low concentration to where they are at a higher concentration. So you can kind of think of passive transport like going downhill and skiing down or sledding down a nice hill, and then active transport is carrying that sled back up the hill to slide down it again. This little lecture is going to focus on our passive transport processes. So we really have two types of passive transport processes. We have diffusion and filtration. So filtration occurs typically across the walls of our capillaries, those really microscopic blood vessels. And it all has to do with kind of filtration pressures and the various kind of pressures exerted on our capillaries. Diffusion is how everything else moves when it moves passively. We have three types of diffusion. We have simple diffusion, facilitated diffusion, and osmosis. So simple diffusion is just a molecule unaided moving down its concentration gradient. To facilitate means to help. So facilitated diffusion is a solute moving down its concentration gradient with the help of either some sort of transport protein. And then osmosis is actually the diffusion of not the solute, but the solvent, the fluid. So let's watch a little video.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across the biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So oxygen and carbon dioxide are small enough that they can kind of slip through kind of in between the phospholipids, so they can move via simple diffusion, passive transport.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So anything that is lipid soluble, or again something small enough like the carbon dioxide and the oxygen, can kind of slip through via simple diffusion. molecules that are insoluble in lipids, molecules that are polar, or big kind of clunky molecules, require some sort of facilitated diffusion. Now, those substances may move in or out of the cell through a carrier protein, which will actually kind of change shape in order to move things across the membrane, or an open channel. So this is kind of channel facilitated diffusion. Typically ions will move through these kind of just always open channels. Now, we do have special types of channels called aquaporins, right? Water pores, and so osmosis is the movement of the water, the movement of the solvent, typically through one of these little aquaporins. Let's look at, so if you have a selectively permeable membrane, when you have a solute that has a higher concentration on one side than it does on the other side of that membrane, then the solute will kind of move down the concentration gradient until an equilibrium has been achieved, the same concentration on each side of the membrane. However, if you have a membrane that is impermeable or selectively permeable, where the particular solute can't move, in this instance it's some sugar molecules that can't get past this selectively permeable membrane, then in order to achieve an equilibrium, since the solute can't move, the solvent has to move. And so in that instance, the solvent will go from the low solute concentration to the high solute concentration. Now the solvent itself is still moving down the concentration gradient, but again we have achieved equilibrium because we may have a higher solute concentration here, but now that we have a greater volume of water, we still have the kind of the same concentration overall across that membrane. So let's watch a little video about osmosis.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid, and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell, and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell, and it will burst open, or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in this shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored.
So tonicity refers to the ability of a solution to kind of change the volume of water inside a cell. There are three types of solutions that we can find: iso, hyper, and hypo. So iso that means equal, so it's the same concentration as the fluid inside our cells. Hypertonic is, hyper means like kind of, I think, hyperactive, right? More. So it's a higher solute concentration than the cytosol. And then hypo means kind of less, below. So it's a lower concentration. So we can either have the same concentration as the cell, a higher concentration than the cell, or a lower concentration than the cell. Those are our only three options, right? Now, plant cells and animal cells react differently to the different types of solutions, as you saw in the little video there. So what our animal cells like are isotonic solutions. So some water moves out, some water moves in, but since the environment has the same concentration as the intracellular environment, the water losses are kind of balanced by the water gains, and the cell does not change in terms of its size. If you place an animal cell in a hypertonic solution, a solution that has a higher solute concentration than the cell, the water moves out of the cell to that higher concentration of the fluid, and the cell will shrink or crenate. If you place an animal cell in a hypotonic solution, where the surrounding solution has a lower concentration than the cell, then the water will come into the cell, and the cell will kind of fill up like a balloon, which could potentially, if it filled up too much, could lead to what we call lysis, which is kind of like the cell kind of bursting open.